Today we're going to talk about acid-base balance. So first off, we have to establish a definition of what is an acid versus what is a base. And for our purposes, the easiest way to conceive of an acid versus a base at this point is to understand it in terms of whether a substance can donate a hydrogen ion to a solution versus absorb a hydrogen ion to a solution. So we call things an acid when it is a compound that donates a hydrogen ion to a solution. And we call things a base when it can absorb or attach to hydrogen ions already in the solution. So if we think of a common acid that is relatively weak, called acetic acid, which you might recognize as being vinegar. Acetic acid, when it's in its acid form, has a hydrogen ion that when introduced into water or any kind of aqueous solution, acetic acid will ionize, detach from that hydrogen ion, and donate it to the solution. So acetic acid, when it ionizes, it becomes a free hydrogen ion and a negatively charged, what we call acetate. Okay. Now, acetate is the form that acetic acid takes when it liberates that hydrogen ion into the solution. So it becomes negatively charged, and what it also is a base at this point. Because if you notice, this symbol here in the middle of the equation means that this equation is reversible. So this process is reversible. So acetate can absorb, as a weak base, it can absorb a hydrogen ion from solution and become acetic acid. And the reason why I'm calling it a weak acid versus a weak base is because acetic acid has a tendency to ionize at relatively high pHs on the acid scale, and I'll define what that means, pH, and how we measure the acidity of a solution. Okay, so strong acids fully ionize even at relatively low pHs, while weaker acids will only be partially ionized at physiological pHs. Okay. And I'm introducing this idea of a weak acid and a weak base because ultimately when we talk about acid-base balance, what we need to talk about are buffer systems. And we'll get there, but all buffer systems are weak acids that have conjugate bases. Okay. So this compound can really exist in two forms: as acetic acid when it is neutralized and non-ionized and is attached to that hydrogen ion, and it can exist in an ionized form where the hydrogen ion is donated and it becomes acetate, which is a negatively charged base. Okay. Now, when we talk about the pH of a solution, the pH is a measure of the concentration of free hydrogen ions in a solution. Now, the pH is actually calculated as the negative log of the hydrogen ion concentration in a solution. Now, what this does, by converting it into a log scale, what this means is that when we look at pH, the full pH spectrum runs from 0 to 14. Okay? That is the full pH spectrum. And then the lower end of the numbers from 0 to 7 are numbers that are acidic. Okay? So 7 is totally neutral. Excuse me. 7 is totally neutral. And as we go down from 7, right, as the number gets smaller and smaller, the concentration of hydrogen ions gets larger and the acidity of the solution increases. Okay? Now that's an important thing to keep in mind because that's something that is easy to forget, that as acidity increases, the pH number decreases. Okay? So 7 is neutral. As you get closer to 0, the hydrogen ion concentration increases, which causes the acidity to increase. And then in the opposite direction, again, 7 is neutral. So as the numbers get bigger, the pH of the solution becomes more alkaline. Okay? So the hydrogen ion concentration gets smaller, and the concentration of what we call hydroxyl ions, or OH negatives, increases. Okay? And we're not going to talk too much about OH negative or hydroxyl ions, but understanding that hydrogen ion concentration increases at a pH number below 7 and decreases at a pH number above 7. Okay? And that is essentially our measure of pH. Now, the pH of body fluids is relatively neutral. Okay, so the pH of blood and cerebrospinal fluid and interstitial fluid and the different fluid compartments, pH of our cells, is relatively neutral. And that's very, very important. The pH of body fluids greatly affects the structure and function of all the proteins in our body, which include all of our enzyme systems. So proteins are very sensitive to changes in pH. Even tiny changes in pH will mean major changes in the structure and function of proteins, which means that all of our enzyme systems that catalyze all the reactions of our body are highly sensitive to pH, which means that we, in terms of the function of our bodies and the maintenance of our health, is extremely sensitive to pH. So the pH of arterial blood is maintained in a range of 7.38 to 7.42. Other sources you might find will, every source that you find will quote something slightly different. Another range that you'll see later on is normal, 7.35 to 7.45. Okay, but somewhere within that fairly narrow range all of our body fluids are actively maintained in that range. Now, how our body achieves this maintenance of pH is both by the employment of buffer systems, which we'll talk about in a second, and then we also have active regulation of at least one particularly important buffer system by both the lungs and the kidneys. Okay, so the lungs and the kidneys, in addition to their other roles in the body, their day jobs, they also have a side job, which is being a primary regulator of our acid-base balance. Okay. So every solution in our body, every fluid compartment in our body, contains a variety of buffer systems. And what a buffer systems are, they are weak acids that also exist in their weak base form. And they, because they are weak acids and weak bases, they can readily either absorb extra hydrogen ions or hydroxyl ions, or donate hydrogen ions in order to prevent any fluctuation in pH. Now, every buffer system, every one of these weak acids and bases are able to absorb or donate hydrogen ions within a range of pH. Okay, so they all have their working range, and the working ranges of all of these buffer systems I'm about to talk about are slightly different. And since they're all present in body fluids, what that does, since they all have slightly different operating ranges, it extends the range of pH that body fluids are able to maintain. Okay, and when we talk about being able to absorb a hydrogen ion or donate a hydrogen ion, when you have a volume of body fluid, so a volume of blood, a volume of cerebrospinal fluid, and if you introduce, let's say, an acid into the solution, okay, that acid is going to donate hydrogen ions. Now, what buffer systems do is that when extra hydrogen ions come into the system within their operating range, this buffer the conjugate bases of these buffer systems are going to attach to those hydrogen ions, absorb them, so that the overall concentration of hydrogen ions and therefore pH of the solution remains the same. So if you add hydrogen ions, the bases of the buffer systems will absorb them. If you take away hydrogen ions or introduce a base, the acids will ionize and donate hydrogen ions to solution, again maintaining the pH. So we have several buffer systems that exist in all of our body fluids. There's one that is particularly important, that's most important, and that's bicarbonate. And if you recall, bicarbonate is the form that we find carbon dioxide in. About 60% of carbon dioxide found in the bloodstream is in the form of bicarbonate. Now, bicarbonate, that term bicarbonate actually describes the base form of this buffer system, which is the HCO3 negative. The conjugate acid is carbonic acid, which is H2CO3. And what you'll find true of all of these buffer systems is that they exist in two different forms. One that's neutral and has an extra hydrogen ion, and one in which they've lost that hydrogen ion to the solution and is now a negatively charged base. Okay? So the acid form is neutral with an additional hydrogen ion, and the base is negative with a lost hydrogen ion to the solution. Okay, so bicarbonate is derived from carbon dioxide, and it is really our primary buffer. Hemoglobin can also contribute some buffering to body fluids. It can actually be found, it's a very large protein, and it can actually be found with a hydrogen ion attached to it or a hydrogen ion unattached to it. Proteins, nearly every protein in our body, both found within the cell or outside of the cell, can also exist as an acid with a hydrogen ion attached, or as a base negatively charged with a hydrogen ion unattached. Phosphate, which is all over the body, including contributing to the formation of ATP, adenosine triphosphate, which is our form of energy. Phosphate also exists as a relatively weak acid and a relatively weak base. Okay. And the interesting thing about phosphate is that even in its acid form, it is still negatively charged. So a phosphate can have two hydrogen ions attached to it or just one. Okay. And that's how it can either absorb or donate a hydrogen ion to the solution. So by far the most important buffer system of our body is the bicarbonate carbonic acid system. And it is the system that the lungs and the kidneys help regulate in order to enhance the ability to buffer body fluids.